Hello and welcome to our video lecture on section 3.2 where we're going to talk about carbohydrates, which would include everything from grains to fruits to vegetables, and that really is going to help provide energy for our body, especially when we can break things down into glucose, which is a simple sugar and a component of starch. Our learning objectives will be to discuss the role of carbohydrates in cells and in the extracellular materials of animals and plants. We'll explain the carb classifications and list monosaccharides, disaccharides, and polysaccharides that are common. Now let's get the molecular structures down. So we have a stoichiometry formula for carbs, and it is CH2O, and in parentheses with the subscript N after it. And that's where the N is going to represent the number of carbons, so we can essentially change the sugars or the larger molecule of carbohydrates. Our ratio will always be a 1-to-1, and that is representation for carbons to hydrogens to oxygens. And our three subtypes are monosaccharides, disaccharides, and polysaccharides. Starting off with our monosaccharides here, mono means one, and saccharide means sweet. So these are our simple sugars. It's just one sugar. You could see a couple of these chemical structures on the right-hand side here, and you'll notice that some of them are going to have the suffix of "-ose," and that will be a clue for us that this is a simple sugar. You may also notice that we have these other terms down at the bottom here, and so if we have an aldehyde group within our sugar, then that is going to be classified as an aldose, and if we have a ketone group in our sugar, then it is going to be classified as a ketose. And then also we can name it based on the number of carbons in the sugar. So if we have 3 carbons, 1, 2, 3, we are going to call this a triose. If we have 5 carbons, then we're going to call it a pentose. If they have 6 carbons, then we call it hexose. And you could see that these are linear chains here, although they can be a ring-shaped type of form as well. Examples of some of the monosaccharides would be like glucose, fructose, galactose. And now we are going to move into disaccharides. So that prefix di represents 2, and it forms when we have 2 monosaccharides undergo the dehydration process, which you could see taking place over here, and we form a glycosidic bond, and this is a covalent bond we find between the 2 monosaccharides. So in this case, we're having those monosaccharides as glucose and fructose come together, and they created sucrose. You can see some other common disaccharides here on the right hand side, such as lactose and maltose. And our last classification are polysaccharides, and these are going to be long chains of the monosaccharides linked by glycosidic bonds. And you could see that we have our versions of unbranched or branched, and our examples include starch, which you can see components of that on the right hand side here. Our plants are going to store these sugars in the form of starch, and so we get these glucose molecules that are all bound together. These 3 are creating amylose, and then we have our 6 here creating amylopectin. And when we consume plants that have these components within them, we have enzymes in our body to break them down, such as amylase. So anytime we see a suffix of ASE, we're going to know that it's an enzyme, and so it comes in and it cleaves these bonds to break it apart so that we can have smaller molecules such as glucose and maltose, so our body can absorb that and utilize it to create energy. Our other example is glycogen, which you may have heard of before. This is our storage form of glucose within our body, and we can store glycogen within our liver or our muscles, and essentially are just repeating units of glucose. So we store this so that when our blood glucose levels start to decrease, we can pull glycogen out, break it down, and release the glucose to get our blood sugar levels back up. And this process is called glycogenolysis. Our next example is cellulose, and we see repeating units of glucose in here, but every other one is going to be flipped. And so this change in conformation makes it difficult for our human digestive enzymes to break it down. But we see that herbivores such as cows, koalas, and buffaloes are able to have an enzyme that can break it down for us. And we find this within a plant's cell wall. Our last example, chitin, is a nitrogen-containing polysaccharide, and we find this within the cell walls of fungi, and also it creates the exoskeleton of arthropods, so like insects, crustaceans, and other insects of that sort. And lastly, we need to discuss the benefits of carbohydrates. When we consume a carbohydrate, 1 gram of it is going to provide us with 4.3 calories. And our carbs can include soluble and insoluble elements. The insoluble part we know as fiber, and that's made up mostly of cellulose, will help promote regular bowel movements by adding bulk to our feces, and then regulates the blood glucose consumption rate and helps remove excess cholesterol from our body. It does this by having the fiber bind to our cholesterol in our small intestines, and by binding to it, it stops those cholesterol particles from entering our bloodstream, and it just exits through our feces. It also helps to do things such as reducing the occurrence of colon cancer, so it has a protective role there, and gives us feeling of fullness. Now, the soluble part, such as glucose, is immediately used as a source of ATP within our cellular respiration processes, which we're going to get into in another chapter.